it's also my privilege to present on behalf of my co-investigators the final overall survival analysis of the TOURMALINE-MM1 Phase III trial of Ixazomib, lenalidomide, and dexamethasone in patients with relapsed and relapsed refractory multiple myeloma. And just to remind everyone, this was a double-blind study which demonstrated a statistically significant improvement in progression-free survival with Ixazomib, lenalidomide, and dexamethasone compared to a placebo combined with lenalidomide and dex in patients with relapsed or refractory multiple myeloma. And obviously, the final survival analysis for such a large, important Phase III is I think, very important to practitioners, particularly as Ixazomib has become a mainstay of all therapy during the pandemic to provide a very convenient and well-tolerated proteasome inhibitor for patients in whom it would be appropriate to think in those terms and or use the drug. And so basically, just to remind people, it's a large study, 360 patients in each arm, in fact, 360 to Ixazomib, lenalidomide, and dexamethasone, and 362 to the control group. And we stratified patients by the number of prior therapies, their previous proteasome inhibitor exposure, and their ISS staging, whether they were 1 versus 2 1 or 2 versus 3, I beg your pardon. And overall survival by intent to treat was a key secondary endpoint. And so with a median follow-up of 85 months, we were able to demonstrate, and I think this was quite remarkable actually, that the median overall survival for Ixazomib RD was 54 months versus 52 months for the control group. And of course, that hazard ratio was 0.939 and therefore, broadly equivalent. But what was particularly striking was the length of median survival that we saw, which was very good news for patients in both groups. And these median overall survival values are actually the longest reported in Phase III studies of lenalidomide and dexamethasone triplets so far in the relapsed refractory setting. So whilst the PFS benefit that we saw with Ixazomib RD didn't necessarily translate into a statistically significant overall survival benefit by intent to treat, what was really interesting is that we looked at OS benefit in subgroups, and we found actually that OS benefit was greater in subgroups with adverse prognostic factors. And that, I think, was an important observation. Specifically, if you looked at refractoriness to prior therapy, that clearly was favorable for the 3 drugs over the 2. If you looked at patients with ISS stage 3 disease, clearly the 3 drugs were superior to the control group. And last but no means least, high-risk cytogenetics, and in particular, not only the classical definition of high risk, but also 1Q amplified patients benefited from the 3 drugs over the 2. So an important treasure trove, I would suggest, of information for practice, not least of which also the combinations were well tolerated, and vitally, there was no difference in the rates of second primary malignancies between the 2 arms, which remain fortunately relatively low in both groups at around 10%. But nonetheless, important to note that there was no new safety signal in that regard. And then I think what was particularly interesting in our analysis is we looked at how, in fact, subsequent therapies impacted on outcome. And what was very interesting there is that there were differences in the amount of antibody exposure. So, for example, in the patients who were on the control group, actually a higher proportion received daratumumab. Also, importantly, in this blinded study, proteasome inhibitors were used. And as a result of that, that may have led to some of the observations of equivalence that we saw. So important to bear that in mind as we think about future trial designs, especially if they're governed in any way by a secondary endpoint, such as a survival endpoint, to be aware of what sequencing of therapy means. So nonetheless, very important data, we think, and it was a privilege to share it on behalf of co-investigators at the EHA meeting. And this was published simultaneously in the Journal of Clinical Oncology as a manuscript, and that went online at the same time as the EHA meeting.